Hello everyone, welcome to our channel that is Funny Life. So let's start the study of life, that is biology. So today we will read plant physiology. The very first chapter of plant physiology, that is transport in plants. So let's start. Transport in plants. In this chapter, there are basically four important topics. The very first is the intro given here in the first page. Next, the means of short distance transport. Next, plant water relations. After that, the long distance transport of water. And the last, the long distance transport of food. That is translocation of food. So, this topic we will be dealing with. In the first page, the main introduction is given in the whole chapter, that how water and food get transported through short distance and through long distance. Unlike animals, even no circulatory system is present in plant, then how the water and food get transported to such long distances in plants? There should be some system present there. Yeah, there is some system, and that is the vascular system, which consists of the xylem and phloem. And this long distance transport through xylem and phloem is known as translocation. For short distance transport, there are some processes like diffusion, facilitated diffusion, and some active transport system. For xylem transport, the transportation, it is unidirectional. It is basically unidirectional. But in case of phloem transport, the transport is multidirectional. It can be uni or bidirectional. Next, from the storage organs, the nutrients are later re-exported. Re-exported means why they are re-exported? Because from time to time, the fluctuation of seasons, due to the fluctuation of seasons, the shore sink relationship also gets extinguished, and this will read further. Next, when any plant goes senescence, nutrients may be withdrawn from such regions and moved to the growing parts. Yeah, this will also be dealing with that which are those nutrients which are mobilized from senescent parts to the growing parts, and which are the immobile nutrients. Okay, and hormones are also transported, some other chemical signals are also transported, but in very small amounts. Next, come let's come to our very important topic, that is means of short distance transport. This is the very first and very important. So the first is diffusion. So I have written here, the diffusion depends on concentration gradient, membrane permeability, temperature, pressure, size of substance, and the solubility in lipids. So this is all where the things in which diffusion depends. Diffusion is passive, it is random and slow process. It is independent of the energy and even not dependent on any living system. Means what? It means that diffusion occurs both in living as well as non-living things. Diffusion in solids is more likely than rather it is more likely than of solids. It means what? It means that diffusion is more likely in solids. Means like gram. If we'll take a gram and put it in water, then after some times the gram, that which is a solid, it will soak water. Diffusion will occur here. Okay, so diffusion in solids occur, but diffusion of solids, it is just negligible. It doesn't occur. It is a very slow process. It is just negligible. So diffusion in solids is more likely than of solids, and it is the only gaseous movement within the plant body. Of course, diffusion is the only gaseous movement. Next, facilitated diffusion. It is another means of short distance transport in plants. So for facilitated diffusion also, gradient should be present already, and it also depends on the size of substance. Smaller substance diffuse faster, of course. Next, solubility in lipids. Whose solubility in lipids? The substance solubility. If the substance is soluble in lipid, then it will diffuse faster. Okay, but in case of hydrophilic moiety, means the substance which are insoluble in lipids, they will be hydrophilic, of course. Hydrophilic means water loving, so it will be difficult to pass for them through the membrane. So for such moieties, for such substances, protein is needed for acting as a channel, and that is the facilitated diffusion, in which there is some channels, and those channels are nothing but proteins only, which carry the substances from one side to other of the membrane. In this case, ATP is not required. This is also energy independent process. Saturation is there, of course. What is saturation? Saturation means that when all the export transporters they are being used, now that is saturation. That is the case of saturation. It means that all the protein transporters they are being used up. And next is specific, of course. Since proteins are used here in this diffusion, facilitated diffusion, so it is very specific. Of course, sensitive to inhibitors also. Proteins are very sensitive to inhibitors. Some channels are always open, others are controlled. Channels are variable in this case. Examples are given. Porins. Porins is also one of the channel. It is also proteins only that form pores in the outer membranes of plastid, mitochondria, and some bacteria. Remember, it is some bacteria. Um, Next example of some proteins are given. That is aquaporins. Aquaporins is what? They're just water channel, and these water channels they are made up of 8 different type of aquaporins. Okay, 8 different type of aquaporins. So symports and antiports. What are these symports and antiports? Symport is the transportation in the same direction of two molecules. Some channels or some proteins are there which allow only two molecules, not only, it's just two molecules. They don't allow only one, they allow two molecules to pass through them, and if two are there then only they will transport. So such proteins they can be symport or antiport. Symport means two molecules in the same direction, antiport means two molecules but in opposite directions. Okay, next is the uniport. Uniport, it is independent of other molecules. Why? Because two molecules are not needed, just one molecule transportation is there. Okay, through the proteins. Next, come to the third means of short distance transport, that is active transport. So active transport is the transport which uses energy, and pump are required. Pumps, what are pumps? They are special proteins which uses energy to carry substances against the concentration gradient. Okay, and against the concentration gradient means not from high concentration to low, but from low to high. That is uphill transport. This is known as uphill transport from low to high concentration. And here also proteins are used. Pumps are proteins, so proteins are used. It means saturation is there, and they are also very specific and even sensitive to inhibitors, right? Here is a comparison given between the three means of short distance transport. So proteins are responsible in case of facilitated transport and active transport. So of course here special membrane proteins will require, yeah, highly selective it will be, transport will get saturated, even inhibitors sensitiveness is also there in case of proteins. Okay, and in case of some diffusion there is nothing required, even no energy, no proteins. Okay, no saturation, no selectiveness, no specificity. Okay, diffusion is very simple, random, and very slow, but slow process. ATP is required only in active transport. Uphill transport is also in just active transport. Next, come to a very important topic, that is plant water relation. So in plant water relation, a plant has a very close relation with water. You know how close? Because the protoplasm of the cell now means cell protoplasm. It means what? The inside of the cell. The inside of the cell is filled with protoplasm only. So that is nothing but water only. You know, examples are given here that watermelon, it has over 92% of water only. And most herbaceous plant, they have about 10 to 15% of their fresh weight as dry matter. It means what? It means that 85 to 90% of the fresh weight of most herbaceous plant is water. Fresh weight includes both dry weight as well as wet matter. So 85 to 90% is water only in most herbaceous plant. Okay. Next is plants, most not all plants, they leave the laws. They lose water through transpiration. That's why it is the basic requirement. Water is the basic requirement in all the plants. Next example is given here. See that the corn plant absorbs almost 3 liters of water in a day. A mustard plant absorbs water equal to its own weight in just about 5 hours. You see how much important water is, and it is of course a limiting factor for plant growth. It can become in the coming days a very important limiting factor it is. The next important topic is water potential. So what is water potential and which factors it depends upon, and what is the unit of this, what is the symbol for this? Okay, so let's read all this. So water potential symbol for is psi. It is given here, the psi, the Greek symbol psi, that is psi. It is expressed in pressure units like pascals. It is a symbol for water potential. And solute potential and pressure potential, these are the two components of water potential. The relationship is also given here in the next page, that water potential is the sum of solute and pressure potential. So what is basically water potential? Water possesses kinetic energy, okay, and due to that kinetic energy, water has some potential. And if the kinetic energy is greater, then the water potential is also be greater. So how could be the kinetic energy greater? For that, there should be more water molecules, and the more the water molecules, the more will be the water potential. Okay, so the greater the concentration of water in a system, the greater is kinetic energy or water potential. So it is obvious that pure water will have the greatest water potential. Next, water molecules, the net movement is from higher energy to the low energy. It is noted by this. Okay, and the water potential of pure water at standard temperature, which is not under any pressure, it will be zero. Why it will be zero? Because it is not under any pressure. Pure water, it is zero, because it has, see, the relation between the relation between solute, pressure and water potential. What is that? Water potential is the sum of solute and pressure potential. If there is no pressure, then pressure potential will be zero. And for pure water, it means there is no solute even. It means that the solute potential will also be zero. Then the sum will be obviously zero. So, in case of pure water, which is not under any pressure, the water potential is zero. I think you have understood. So, yeah. If some solid is dissolved in pure water, then the water molecules or the water potential gets decreased. Why? Because solute potential is negative and pressure potential is positive value. So if we'll add solute, then the water potential decreases. The magnitude of this lowering due to the dissolution of solute is known as solute potential. It is always negative. Okay, and if we will further add the solute, now it will increase the solid potential. It means the solid potential gets more negative. More negative means mathematically it means less. Magnitude will be less. More negative means less magnitude. Okay, so next there is the relationship between pressure and solute potential and water potential given in the next page. Let me show that. What is that? Yeah, here it is. Yeah, here it is now. Okay, here the relationship is given. Water potential is equals to psi s plus psi p. This is the value. And pressure potential is always positive, though in plants, negative potential or tension in the water column in the xylem plays a major role. So pressure potential is positive and solid potential is always negative. Pressure potential is denoted as psi p. Okay, fine. Now come to osmosis. I think you have understood about the water potential. What is that? What relationship is there? What does it depends on? Okay. So osmosis, it is also a type of diffusion only. But in this case, in this case, the osmosis depends on both pressure as well as concentration gradient. But diffusion depends just only in concentration gradient, not in pressure gradient. Okay. Osmosis is a special type of diffusion. Imbibition is also a special type of diffusion. Here is a very important line is written that the cell wall, it is freely permeable to water and substances in solution. So cell wall, it is not a barrier to movement. Okay, cell wall is not a barrier. Next comes the cell membrane. So the cell membrane and the vacuole or the vacuole membrane known as tonoplast. Vacuole membrane is known as tonoplast. They both are very important determinants of movement of molecules in or out of the cell. This is a very important line. Okay. Osmosis is the term used to refer specifically to the diffusion of water across a differentially or selectively permeable membrane. You know, the net rate is dependent on pressure and concentration gradient. Okay, this is fine. Now question is given here, solution. Yeah, see the figure is given here. In this case, you have to say solution of which chamber has lower water potential. So I have written here B. Of course, it has more solid molecules. It means low water potential. Okay, more solid molecules, less water molecules. That's why low water potential. Next, which has lower water potential? Okay, which has lower solid potential? It will also be B. Why? Because lower solid potential means lower solid potential. It means less negative value of solid potential. So less negative is where there is less number of solid. Okay, less number of solid is in A. Okay, so there will be less negative means high solid potential, and when where there is more solid molecules, then there will be high solid potential. Okay, so here the answer will be B. I think you have got it, that why B is there, because in this case there are more solid molecules, and since there are more solid molecules, the value of solid potential will be more negative, and since it is more negative, it means the value will be low. So B has low solid potential. Okay, which solution has a higher solid potential? It is low, it has lower, it means A will be higher solid potential. In equilibrium, which chamber has a lower potential? Both will have equal potential. In equilibrium, both will have equal. So this one, F number, C, F number also 2000 kilopascal and another has minus 1000. So minus C, when there is minus now, then the value which is smaller, that will have the value which is smaller, that will have greater magnitude. Okay, so it has having greater magnitude, so minus 1000 kilopascal, it has the higher water potential. Next, what will be the direction of the movement of water when two solutions with this 0.2 megapascal and 0.1 megapascal? The direction will be, of course, from higher to lower, so 0.2 to 0.1. Okay. So experiment, it is given about osmotic pressure, and osmotic pressure, yeah, membrane is required. So here is a process to make the membrane. What is that process? First of all, make a hole in the egg and then place the shell in a dilute solution of hydrochloric acid for a few hours, and after that the shell will dissolve and you will get a membrane. So this is the process of taking out a membrane, semi-permeable membrane. Okay. Next, the pressure required to prevent water from diffusing. It is only the osmotic pressure, and this osmotic pressure, it is directly proportional to solute concentration. Okay. So osmotic pressure is a positive value but directly proportional to solute concentration. See here written also, a solid osmotic pressure is a positive value but osmotic potential is negative. So osmotic potential is just like solid potential, nothing but solid potential, and osmotic pressure, it is just like pressure potential. Okay. So that's the last topic. No, not the last. It is plasmolysis. Plasmolysis, what is that? The solution. The solution is here. It can be hypertonic or isotonic. So what is in case of isotonic? Actually, in isotonic, the external solution balances the osmotic pressure of the cytoplasm. It means the both the external solution and the inner solution, means the inner cell, both have the same pressure, that is the same potential actually, that is isotonic, equilibrium is there. And hypertonic, in case of hypotonic, it means that the solution, external solution, is more dilute, and if it is more dilute, then what happens, the cell will go inside, sorry, the water will go inside the cell will swell up. So this is hypertonic solution. And if the external solution, external, the outer solution is more concentrated, means less water molecules, then of course the cell will shrink, and this is the case of plasmolysis. This hypertonic condition only leads to plasmolysis, okay, and leads to the formation of a plasmolyzed cell, that is the shrinkage of a cell away from the cell wall. Okay, these are the photographs, I mean the diagrams. Diagrams are there for plasmolyzed cell, flaccid and turgid. In turgid, in case of turgid, there is a turgid pressure against the cell wall. There is a pressure. There is a very important question, that what occupies the space between the cell wall and the shrunken protoplasm in the plasmolyzed cell? So the answer will be not water. It will be the external solution. The external solution in which the cell has been placed. That solution occupies the space. Okay. In case of plasmolysis, what happens, that water moves out first from the cytoplasm and after that from the vacuole. So this is also a very important line. Next, isotonic solution, there is an equilibrium condition, and in that condition the cells are said to be flaccid. Okay. Plasmolysis is reversible. So once the cell gets shrinked, after that it can be regained also. Okay, it can regain its shape by putting it in a hypotonic solution, means a higher water potential solution. Okay, by this way the cell can again swell up. Fine. So yeah, imbibition. Next topic is imbibition. Yeah, one question is there, what would be the water pressure potential of a flaccid cell? Guess. Think what would be the pressure potential of flaccid cell? It will be zero, of course. Why zero? Because there is equilibrium, no pressure is there. Next, which organism other than plants possess cell wall? Which organism? There are various organisms like fungi. Fungi also have a cell wall made up of chitin. So this possesses cell wall. Next is imbibition. This is our last topic, I think, probably of today's. After that, we'll read tomorrow. Okay. Imbibition is a special type of diffusion. It is also a special type of diffusion when the water is absorbed by solids, colloids, and due to this there is an increase in volume. Example of imbibition are what? Absorption of water by seeds and dry wood. Okay, these are the examples, and applications are to split the rocks and boulders. Germination of seed is also application of imbibition. Without imbibition, the seed would not have germinated. Okay, so imbibition, it is water movement only. Yeah, a longer concentration gradient. In addition, for imbibition, the adsorbent and adsorbent. What is that? Adsorbent and adsorbent means, adsorbent means the surface in which the liquid is imbibing. Okay, the imbibing surface, it is known as adsorbent, and liquid is known as adsorbent, bent, bent. Okay, so affinity should be there between these two. Next is the long distance transport, and this will read in the next part. Okay, so have fun and enjoy the video.